Good morning, distinguished guests, ladies and gentlemen. Welcome to the Tang Prize Taiwan Biotechnology Forum, mRNA, the new generation of medicine. Before the forum begins, we would like to invite everyone in the audience to take a group photo together to remember this special event. So our photographers will take the photo from the stage. We invite everyone to remain on your seats and don't forget to smile. Okay, everyone, please look into the cameras in the center together and smile. Okay, so the camera on your right and the cameras on your left. Beautiful. Okay, once again, the cameras in the center. Thank you very much for joining the group photo. Thank you, that was beautiful. And now we would like to invite board member of the Tang Prize Foundation, Dr. Yijuang, to give us the opening remarks. Ladies and gentlemen, please join me in welcoming to the stage Dr. Yan.
Welcome everyone, ladies, gentlemen, laureates, and panelists. On behalf of the Tang Prize, I would like to give everybody the warmest welcome, especially the weather condition today. Yesterday, we learned a great deal of the knowledge about the new milestone and the new medical era in mRNA. Today, we have an open forum to invite everybody to further discuss about mRNA, especially in diagnostic, medicine, treatment, and the future. Here, I also like to invite our Prime Minister, Dr. Chung-hsiun, to give the opening remarks through the video. Please.
Dr. Yan, distinguished guests, ladies and gentlemen, good morning. On behalf of the Executive Yuan of Taiwan, I am delighted to welcome all of you to this forum. I would like to extend an especially warm welcome to the 3 laureates of the 2022 Tang Prize in Biopharmaceutical Science, Dr. Katalin, Kariko, Dr. Weissman, and Dr. Pieter, Cullis, who have traveled to Taiwan to be with us today. Last year, the 3 laureates in biopharmaceutical science for the discovery of key vaccinology concepts and approaches leading to the successful development of messenger RNA-based COVID-19 vaccines. The new platform they developed is a nucleoside-modified messenger RNA vaccine delivered by lipid nanoparticles. Their breakthrough discoveries and ingenious approaches were key to the rapid and successful development of the SARS-CoV-2 vaccines by Pfizer, BioNTech, and Moderna. Today, messenger RNA technology has become a hot topic in the global biotechnology and medical fields. This technology has demonstrated significant value in vaccines, cancer treatment, and gene therapy. The Taiwanese government understands the tremendous importance of the development of messenger RNA technology, is offering support through various means. The National Science and Technology Council and the Ministry of Economic Affairs are the 2 main governing bodies implementing projects that promote collaborations between academia, industry, and government to facilitate the rapid advancement of this technology. Additionally, the Executive Yuan provides support and investment in establishing contract development and manufacturing organizations, or CDMOs, to align with international standards and implement the practical, translational medicine application. The government is at the same time providing research funding and facility support to strengthen collaboration between academia and industry, nurturing a more robust research ecosystem to elevate Taiwan's capability in this sphere. We believe that it is essential for government, academia, and industry to work together if we are to achieve the long-term development of messenger RNA technology. Furthermore, international cooperation is also important to drive messenger RNA technology forward. Collaborations based on mutual sharing and reciprocity between countries will accelerate the development of messenger RNA technology and maximize its potential. And Taiwan is also working towards this goal. In this era of new possibilities, we believe that messenger RNA technology will bring even more benefits to humanity. Taiwan possesses excellent talents in research and innovation, offering immense potential in the field of biotechnology. Let us work together to facilitate the development of messenger RNA technology and inject fresh vitality into Taiwan's growing presence in international biotechnology. Last but not least, I would like to extend my warmest congratulations and gratitude to the 3 Tang Prize laureates. Their outstanding achievements and contributions have had a momentous impact on human society and saved millions of lives. Thank you for your dedication and efforts. Let us join together to bring to life the many new applications of messenger RNA technology and further contribute to the health and well-being of all humanity. I sincerely wish for the success of this interesting forum and hope that every esteemed guest will gain valuable insights. Please enjoy your time, stay healthy, and best wishes to everyone. Thank you very much.
Thank you very much to Premier Chern, and thank you once again to Dr. Yan. Let's now bring up to the stage our distinguished panel. First of all, please welcome our moderator, Dr. Chung-hsiun, President of Development Center for Biotechnology. And next, we would like to welcome our panelists, 2022 Tang Prize laureates, Dr. Katalin Kariko, Dr. Drew Weissman, Dr. Pieter Cullis, and next, please welcome Dr. Wang Academician of Academia Sinica, Ms. Ho Jinglan, CEO of TTY Biopharm, and Dr. Chen Yijuang, Deputy Minister of National Science and Technology. Thank you very much. Please take your seats. And now let's hand the floor to our moderator, Dr. Wu.
Good morning, 3 laureates and distinguished guests. I think first of all, I would like to excuse ourselves because of the audience. In the audience, we don't have too much audience, but I think today we have a typhoon emergency. Typhoon is just like a cyclone in Australia or a hurricane in America. It could be very devastating. So because of the typhoon is actually here, So the government actually take the precautious the measure to have to call this day emergency day. So today all the country activity in all over the country is actually called off. So not a lot of people here, but people come here as the big fans of the Tang Prize Laureate forum. So please give ourselves a big hands, okay? Thank you. So we have the Tang Prize, the laureates, the presentation already yesterday and the day before. But however, today's forum is open to the public. So a lot of information has been provided, and today we are going to extend more information about the technology in the messenger RNA as well as the LNP, the application. And so I think what I'm going to do is just I have invited the three of our laureates to provide basic information about the contribution in this or induction of this Tang Prize with about 3 to 5 minutes, and then three of our domestic or local very distinguished scientists to express or to introduce themselves again with 3 to 5 minutes. So to begin with, I will just use the order of the seat. And the first speaker is Dr. Katie or Kariko. And yes, thank you. And Kariko, as she is going to tell you that she's a very, very persistent scientist. Actually, all three of our scientists are very persistent scientists. They insist on their interests and keep talking and keep put all the efforts through their career for a very long time. And actually, even in the beginning of their career, some of their mentors or advisors tell them, why don't you do something useful? But it's not because it's not useful. It's because the time is not due yet. And as all of us can see, that's when it becomes very important. It becomes very, very important. It saves the life of at least 2 billion people all over the world. So first of all, I would like to invite Katie, spend about 3 to 5 minutes, explain, say something to us. Okay, thank you, Katie.
Yes, thank you. So I am originally from Hungary. I was born there. I get my education there, my PhD. As an undergraduate, I work with liposome, and as a graduate student, I synthesized short RNA. Those were not messenger RNA, those were just 3 nucleotides, but at that point it seemed that it was an antiviral compound. And so we tried to develop to have something helpful things to do, because my supervisor came from the pharmaceutical industry and he wanted to make it something that it is helpful. So I was working with RNA, and then I moved to the United States 1985. Again, I still working on RNA, short RNA, this short molecule, 2' 5' link antisense molecule. And then we also started to do long double-stranded RNA molecules, and those induce interferon. And we started already clinical trials. In 1986, we treated HIV patients because that was the major problem in the 80s. And this double-stranded RNA, we expected that will induce interferon in the patient, and the interferon, which interferes with the viral replication, would help them. But unfortunately, it didn't help. But from 1989, when I moved to the University of Pennsylvania, I started to work with messenger RNA. By that time, we could synthesize in a tube, and so it was about like 8 years I spent with the cardiology and the neurosurgery department to figure out how we could use mRNA therapy for this medical field. And not until met my colleague, Drew Weissman, and together started to work, realizing that the messenger RNA, which I was synthesized, was immunogenic. And we did not set out to make non-immunogenic RNA. We didn't know that such things can be done. We were just basic scientists who were curious. And we wanted to understand why it is immunogenic. And when we analyzed different RNA and found that one type of RNA which had a lot of nucleoside modification was not immunogenic, we asked the question that whether we can make a messenger RNA containing these modification, one of them, all of them, and whether such RNA could be translatable. So it was always a basic question we asked, basic science, and not setting up something. But when we found that we can make RNA non-immunogenic, then we proceeded, and that's we are here today, mainly. Thank you.
Okay, thank you. And the next one is Drew Weissman. And Drew used to work with the legendary figure, Tony Fauci. And as he told us yesterday that in one time Tony asked Drew about why don't you do something useful, and actually turned out the achievement of Drew show that Tony was wrong. So the lesson learned here is that even though with the legendary figure in science, if you still have to insist on what you believe in, and then if you persist on what you believe in, then you eventually success. So please, Drew, 3 to 5 minutes. Thank you.
Thank you. So just to note, I've never said that Tony Fauci was wrong. So I grew up in Massachusetts. I went to medical school and did an MD, PhD. Was always interested in being a physician scientist. I did my fellowship in Tony's lab and studied HIV research. Tony had one thing that we were not allowed to do. We could do anything we wanted. We could study any aspect of HIV immunology, but he didn't want us to develop vaccines. So when I moved to the University of Pennsylvania, as an impetuous child, the first thing I wanted to do was work on vaccines. So when I got there, I started to figure out how to do that. And I had studied dendritic cells in Tony's lab. That was something brand new. There were only a couple of labs in the world that could make dendritic cells and study them. But I knew that dendritic cells were the most potent antigen-presenting cell. They were the cell you had to target with the vaccine. So when I got to Penn, I started to search for ways of loading dendritic cells to make a vaccine. And we had viruses, we had DNA, we had peptides, we had proteins. We didn't have RNA. And that's where I met Katie at the photocopy machine. Most people here don't know what that is and have never used one. But in the old days, that's how you read papers. So Katie and I started to talk, and she told me about how she made RNA. So we started to work on vaccines. And that's where Katie described our journey, finding RNA was immunogenic and how to not make it immunogenic. But I kept up with the vaccines through that entire time. And that led us to where we are today with the COVID-19 vaccine. But during all of this time, my passion was in equity, in allowing the entire world, including low-income countries, access to this technology. And I talked about this yesterday. I've built 18 GMP sites around the world that can make RNA for clinical development. And I've set up research infrastructure across the world that studies RNA. And what that does is it allows local communities, local countries to develop vaccines and therapeutics that they need, that big pharmaceutical companies have no interest in developing. So we're now developing vaccines for dengue and tularemia in Thailand, for house dust mites in Vietnam, for malaria and a variety of parasitic diseases in Africa. And we continue to expand this applicability and this ability for local countries, communities, regions to develop their own therapeutics. Thank you.
Okay, thank you, Drew. As you can see, is that the scientists could not only do business, but at the same time can be conduct many activity which has fit the high humanity. In this case, delivering the medicine to the ordinary people at the economic cost is very important, and I think that is the belief of the Drew. Okay, thank you. And the next panelist will be Pieter Cullis, and Dr. Cullis, he is also told by his mentor that why don't you do something important instead of doing some oil? But the thing is, in the lipid nanoparticles, the field, the company that he found, the Cullis, and also the machine which is very important for making lipid nanoparticle, and it's made by the company called the Precision Nano System. Both of the company are founded by Pieter, and he is trained as mechanical engineer, chemical engineering, right?
Physicist.
Or physics, and he's very handy. So all of the machine that he used throughout his research is actually either made by himself or designed by himself. And he's a very creative person and also very entrepreneur. So as I just say, Cullis and precision medicine are founded by him. So please introduce yourself, Pieter. Thank you.
Thank you. Yeah, the journey that I've been on is really a you know, as you've already heard, we've all gone through different situations to be here. I started off my academic life as a physicist, and so I ended up getting a PhD in physics, in solid state physics. I was looking at semiconductors at 4 degrees Kelvin. This is like looking at transistors on the moon. Anyway, very difficult to explain to my girlfriend, so I thought I should take a different tack. And so this is where I made a decision to move into the life sciences, and I knew a bit about nuclear magnetic resonance, probably more familiar to you as MRI, but and so I decided to try and apply that in life science research. So, but I'd never taken a course in biochemistry or in molecular biology, and I still haven't actually. I've taught those courses, I just haven't taken them. So this was what I was very lucky. I was in Oxford as a postdoctoral fellow, and I met an individual named Ben de Kruif there, who was interested in lipids, the lipids that are found in biological membranes. These are not the lipids that are found in fat. They're a different class of lipids. And so we ended up studying these using magnetic resonance as the tool. And I found I got more interested in the lipids than I was in the NMR, in the magnetic resonance technique, and so started to move quite dramatically into, okay, well, what are the physical properties and functional roles of these lipids and membranes, because there's literally hundreds. In any given membrane, there's literally hundreds of different lipids there, and we still don't really understand what they do. Well, that's the thing was you couldn't really study the properties of these lipids in the intact membrane because there was too many there. It was too complicated. So you had to make what we called model membrane systems. These are very small. They're very much the same size as lipid nanoparticles. And what we found was we could take these model membrane systems and we could encapsulate cancer drugs in them. And so, of course, this gave us the idea that perhaps we could package cancer drugs and deliver them more specifically to where they're needed, as opposed to going all over the body. And that was in the 1980s that we discovered this. And so we started a company to try to develop these as cancer therapeutics. We're actually quite successful. We had 2 products that have been approved by the FDA and the European Medical Agency. But the follow-on from that was that we moved, said, okay, we can deliver these small cancer drugs. Maybe we can deliver nucleic acid-based drugs. So this was in the 90s, and this was more difficult. And we had a number of hurdles that we had to overcome. But what we found was that we could take what are termed cationic lipids, in a particular class of positively charged lipids called ionizable cationic lipids, and we could use them to encapsulate nucleic acid polymers into these lipid nanoparticles. And one of the advantages of these systems was that they were nowhere near so toxic. In other words, we didn't kill the animals when we injected them into say mice, as compared to using some more standard formulations. That really was a seminal discovery, because not only did it allow us to get more, to encapsulate nucleic acid-based drugs such as RNA-based drugs in lipid nanoparticles, but it also turned out that by varying the chemistry of the ionizable lipids, we could get much more potent systems in terms of delivering that to the interior of a cell. And then a third component, which we had no idea about, was that they turned out to have a really good adjuvant effect in terms of vaccines. And so these the discovery of the ionizable lipids, which we originally developed for very, very basic studies, trying to understand some properties of lipids and membranes, turned out to be very useful for the delivery aspects. Now, just one other point: the only reason we managed to do most of this was in the middle 80s. I had in the early 90s. I had a very, very good academic group, and I wanted to keep them together to try to push these applications forward. And so we started a company then, and this that made the lipid nanoparticles, and we were actually in the cancer field and then in the gene therapy field. And that is really, as you were indicating, that's really been a big part of our success. Is you can do a certain amount in academia, but you can't really develop products in an academic setting, and you have to have dedicated teams in an industrial setting that can really push something forward, and with more intensity. And I think that's been a large part of the reason for any success we've had in this area, is because we've managed to keep the core group of people in an industrial setting together to develop these products. And I think it's an important thing, probably for most jurisdictions, to do this.
Okay, thank you. I think that lesson learned here is that if you want to do the business in science, you have to dedicate it to the business and have a dedicated devotion to it. You cannot do it on the side. Okay. So the next panelist is Dr. Wang, Andrew Wang, and he was the ex-vice president of the Academia Sinica, and he is also a renowned structural biologist. And I'd like to have Andrew to give us some of his opinion, and with very insightful yet very humorous comments. Andrew?
Okay, thank you. According to the introduction here, I should say a little bit about myself as well as research interests. So I'll say a few words about them. I was born in Taiwan and educated in National Taiwan University as a chemist. So as in the 60s and 70s, many of us say, come to NTU, go to the United States. And that's most of us did at that time. Okay, so I went to the University of Illinois, got my PhD in chemistry. And I found chemistry is interesting, but biology is probably better. So I went to MIT to study with Alexander Rich. Okay. So initially I studied tRNA. So I know many of those modified nucleosides at that time, especially in tRNA. There's a loop called T side C, the pseudouridine C. Anyway, so I learned quite a bit about nucleic acid. But later on, I work, of course, in Alex's lab, discovered the zDNA, and many of you may not be familiar with. But zDNA at that time, it's a left-handed DNA double helix, so we thought it's interesting, but we don't know, or we didn't know anyway at that time how important it was. And most of them at the beginning thought that was just a chemical curiosity. But Alex and I, we didn't give up, continue, mostly Alex, to pursue the biological relevance of zDNA. But that was in 1979. So now it's more than 40 years later. I think there are some lights gradually emerging that the zDNA may be important biologically. For example, in antiviral mechanism, when the DNA or RNA enter the host through those pathogens, and it activates an activity called ZBP, the zDNA binding protein mechanism to counter this kind of infection. So zDNA may be important, but it's also discovered that in some enzyme component, there are zDNA binding fragment in there, like the ADA, the adenosine deaminase system. So we were sort of gratified that we can discover something interesting, although at that time, maybe many people doubt about it. But after that, I moved back to University of Illinois as a faculty, continued to my structural biology work. And then in the year 2000, I returned to Taiwan. And as Herbert mentioned, that I worked with President Wang as his vice president at Academia Sinica. So learned quite a bit about the importance of science in Taiwan. So we help. And one of the important things at that time we initiated was to establish the National Biotechnology Research Park. So in short, it's called MBRP, right? And so MBRP now has been kind of established as one of the branch. It's a cross-departmental institution, including DCB is located at the MBRP. And it's now one of the focal points of biotechnology in Taiwan. It's a very important aspect. And later on, I think when we discuss about the role of Academia Sinica as well as MBRP in the messenger RNA technology development, I will elaborate a little more. So I think now I'm pretty old. I think I'm older than most of you. But anyway, so I moved to Taipei Medical Universities as a chair professor. But on the side, I also help quite a bit of companies in biotech as well as in biopharma. So hopefully I can contribute to promote a little bit about biotechnology in Taiwan. Thank you.
Okay, thank you, Andrew. So next is Sara Ho, and she's the general manager of the leading liposome company in Taiwan, and actually has many license agreement with the many different pharmaceutical companies using their technology, and she's very experienced and renowned entrepreneur as well as a manager. Please, Sara.
Yeah, thanks for Dr. Wu's introduction. I just relocated back to Taiwan last year, so I think most people didn't know me. I'm Sara Ho. I was graduated from Taiwan Medical University and Taiwan National University in pharmacy. Before joining TTY, I worked in multinational company for more than 30 years, mostly in Novartis, and specialized in oncology and immunology. I also worked in different countries, such as Taiwan, China, and the APAC regions. So I launched the first target therapy, small molecular, in Taiwan long time ago. So therefore, I have involved in early medicine development and understand how many resources are needed through the cross-team and the platforms collaborations across countries. And I have launched many products as well and faced many difficulties. Sometimes the new medicine use very breakthrough mechanism or just newly identified biomarker. And but the medicines passed the phase 1 and the phase 2 trials with the flying colors, but the main still failed in phase 3s. But now, good news is that we have so many modern technologies like automation, big data, and AI that we can use to expedite all these processes like drug screening and clinical trials. That provide us very exciting opportunities to revolutionize the R&D processes for the new drug development. So I'm really looking forward to the new perspectives and the new development. That is why it's so intriguing to work in biotech industry. Thank you.
Okay, thank you, Sara. So the next one is Yijuang, and Dr. Yijuang. She is also a fellow in Academia Sinica, but currently she's taking the government officer's position in the government officer position, and so well, Yijuang, please. Thank you.
Distinguished guests, ladies and gentlemen, good morning. My name is Yijuang Chern. I'm a neuroscientist in Taiwan working on developing drugs for dementia. I was really deeply encouraged by the life stories of our guests because in the past few years, I've been told, don't work on drugs on Alzheimer's disease, it's too difficult. So thank you for the encouragement. I'm here, the reason being because I am also serve as the Deputy Minister of National Science and Technology Council, NSTC. Now, the most well-known mission for NSTC is that we support basic research in Taiwan each year. We support close to 9,000 projects under the categories of nature science, life science, engineering, technology, and also humanity and social science. Of course, that we are also responsible for the establishments and developments of science park, which serve as an ecosystem to facilitate the technology transfer in innovations. Now, the most important mission of NSTC is to formulate the science and technology policy at the national levels. That's why we've been really pay close attention to emerging technologies such as mRNA technology platform, focusing on two specific aspects. One is, of course, national security for the need of vaccine productions. Another is for industry development. Now, we're very excited about these developments, and thank you for inviting us. Thank you.
Okay, thank you. So we have the first round of introduction, and we know the background of the panelists and all our panelists. So next, we have a specific question for each individual panelist. So the first question is for Katie. Dr. Katie is enough. Yeah, I think it's more sweet. Yeah. Yeah. So based on your your presentation yesterday, I think the use of pseudouridine in the application of messenger RNA, so that the pseudouridinated messenger RNA can be translated and be stable and injected into the bodies and subsequently induce the desired function, such as vaccination or as a therapeutic agents. So please, because of as I think as you mentioned in your talk, there are many different modified nucleoside. So could you elaborate a little bit more about how you discover pseudouridine is the molecule to be used in the messenger preparation of messenger RNA? Please, Katie.
Yes. So in our body, there when we make mRNA or any RNA, actually is always from the 4 basic nucleotides we are synthesizing. And when an RNA is made, or during the process when a messenger RNA is made, there are different enzymes can introduce modification, and there were more than 100 modifications were identified already, which is present in RNA. And in the heydays of modified nucleoside, actually was the 70s when they discovered that the cap part of the RNA has this modification, and the tRNA, which we just heard, tRNA had all of this modification. But you could see that more publication was there, and then it was like less, because somehow they could not have the phenotype. So they changed and they couldn't see difference, and that's difficult to investigate. But so when we realized that the transfer RNA was not inducing any inflammatory molecule when we added to the human dendritic cells in a form lipid, positively charged lipid, we did. So we thought that maybe one of them or all of the modification is needed. We didn't know that. And the enzymes were not known how to introduce them, so we couldn't order from Promega that send those enzymes, and we just modify our RNA, so that we had to order those modified nucleoside triphosphate and incorporate to the RNA. Anyway, we didn't know which one is important, or all of them, or we didn't know many things. But that's how science works. So you don't know, and you are performing experiment, and we are researcher. I don't know whether you ever thought about the word. We are not searcher, researcher, because we are repeating and repeating the experiment. So that's what we were doing, and we just could check out from the 10 of them, the 100 modification, because that was commercially available triphosphate form. And so we crossed our fingers that, hopefully we find one of them or all of them would be important for that. So again, nobody ever did the modified nucleoside with mRNA, so we did that. And turned out that whenever uridine was modified, so not pseudouridine, but 5-methyluridine and 2-thiouridine, all of the uridine modification resulted that the RNA was not immunogenic. So, but of course, when we looked at the translation, so such RNA containing modified uridines, turned out that when it was 2-thiouridine, we get no protein. So that would not be useful, not for vaccine and not for therapeutic protein production. If you don't get protein, you get a non-immunogenic mRNA, but useless. But 5-methyluridine actually translated as well as the conventional uridine-containing RNA. But what we emphasized yesterday, and we say again, the surprise was that pseudouridine-containing RNA translated 10 times more. 10 times more protein was produced. And we could see and spend 2 years, isn't it, to figure out why we get more protein from it. So we could see that it was more stable, and we could detect like 3-4 days the translation from such RNA. And so this gave us hope, that it will be protein produced such RNA will be sufficient for some therapeutic RNA to use So the protein, which, we are not talking about the protein is really the drug. We are delivering the mRNA. So the mRNA we deliver and the protein is which exerts its effect, and the protein half-life can be different. So even the RNA is gone at 3-4 days later, is not there, but the protein half-life could be, a couple of weeks or months. And did I answer your question?
Yeah, sure. Okay.
Thank you.
Yeah, thank you. I think from her story, she has to pass one hurdle after another, hurdle after hurdle, hurdle after overcome. So it's just like I think Katie mentioned that she received many awards, but the one thing that she most proud of that is she has a daughter who actually is the 2-time Olympic winner. And so that's just like the daughter is just like her, one hurdle after another hurdle, and eventually win the Olympic gold medal. Okay, so the next question is for Drew. And Drew, I think you inventory about many product or many target disease that you are targeting. And so could you share again with the audience here, and maybe you can give some direction to the researcher who are interested in going this direction. Thank you, Drew.
Thank you. So, as I mentioned in my introduction, equity to me is important. The way gene therapy is done nowadays is a patient is admitted to the hospital, you take the cells out that you want to change, you put them in culture, you infect them with lentiviruses. All of this is very expensive, very complicated. It can only be done in a couple of centers around the world, and it costs millions of dollars per dose. That doesn't do the world much good. So our interest has been, how do you deliver the therapeutics in vivo? How do you simplify their delivery? And as everybody who's ever worked with nucleic acid or viral therapeutics knows, targeting the right cells in the body is very difficult. So that's what we studied, and that's what we developed. We've been able to target many different cells and tissues. We can target brain, lung, liver, the heart, T cells, CD4 positive cells, bone marrow stem cells, and we continue to increase this. So what this targeting does is it allows you to deliver the therapeutic, and whether that's a protein to treat a disease, a protein to replace a deficient protein, or a gene editing enzymes to fix broken genes, now we can do all of that in vivo in the body. So instead of $4 million to fix a broken gene, we can inject RNA LNPs that target the right cells, bone marrow stem cells, T cells, cardiomyocytes, the brain, to fix whatever the pathology is in that organ. So now it's easy, it's simple, and it's inexpensive to treat these diseases going forward. And to me, that's the most important thing. A therapy isn't very useful if it costs $4 million and insurance companies and governments won't pay for it. You have to make the therapy effective and easily given to patients for it to be useful for the world.
Okay, thank you. I think for your information, we totally agree with you about the medical equity to all population all over the world. And this was started in Taiwan. Taiwan, we have a national health insurance, so every single citizen can enjoy the same quality of medical treatment. So that's the first step in the equity in medical equity in Taiwan. So the next question is for Pieter. And as we all know that the so-called central dogma, we have a DNA and transcribe into RNA. RNA translate into protein. And protein is supposed to be the one who just conduct the activity. But we miss two other major category of the molecule. One is the sugar. Sugar could be very, very complicated. And the other is oil or lipid. And lipid, as Pieter just mentioned, that you have experience or experiment many application of a different type of the lipids, including ionized lipids. So the current formula of the LNP is very useful and has been used in billions of people. And is there any drawback that need to be improved? And is there any future or any directions possibility in finding new lipids in the application of the lipid nanoparticles? Please elaborate. Thank you.
The delivery approach is really in its infancy right now, and as Drew was just indicating, we have the potential for targeting to particular tissues, particular cells in a much more defined manner. Right now, we're more or less confined to vaccine applications going in intramuscularly and to applications that rely on the liver producing a protein that we might want to have some beneficial effect. We're taking a slightly different approach than Drew is. I mean, our objective at the moment is to transfect everything, to have a system where you inject intravenously and then you get global transfection right through the entire animal. And then, of course, then you're going to get into this is a situation then where you're going to want to turn things on and off in particular tissues. And so I think this is, in combination with some of the target, you can only target to things that are already in the circulation. It's difficult to target to things that are not in the circulation directly. And so the approach where we're getting more global transfection, then you're into interesting territory. How do you turn things on and off in particular locations? And so and there's a number of approaches. There's molecular biology approaches to only have messenger RNA that will be transcribed in particular tissues. There's direct physical approaches where we can irradiate from the outside and stimulate lipid nanoparticles to become transfection potent, and so we're working in those areas as well. And anyway, so there's a variety of possibilities that are coming on. There's going to be a number of approaches to getting to the point where we can essentially transfect any tissue in the body and correct whatever lesion, correct whatever disorder is in that region. So we're getting better and better at things like the spleen, the bone marrow, brain with direct injection. We can certainly see remarkable levels of gene expression there. The eye, direct, all of these tissues now are becoming possible to do really very direct interventions in terms of curing whatever disease is localized to whatever region of the body. So I think it's pretty exciting. The really exciting part to me is the rapid nature that you can develop these medicines. So a child is born not making a particular protein, some hereditary disease, these rare diseases. We now have the opportunity to be able to identify what the problem is. Perhaps they're not making a clotting protein. It can be anything. And once we know what is not being made, then we can develop a messenger RNA coding for that protein and package it in a matter of a couple of months and essentially have a very direct way of curing of that disorder. So it's quite remarkable. So this is where the pressure is going to come on in interesting ways in regulatory agencies because they're really not used to these kinds of medicines, which are highly targeted and very effective and very can produce very quickly, but you clearly can't run a clinical trial of thousands of people because you're dealing with just one person that has a particular disorder. And so I think this is going to cause a bit of a revolution, not only from the medicine side, but also from the point of view of the regulatory agencies that have to make sure these medicines are, safe and effective. So we're into some revolutionary times in medicine, that's for sure.
Okay, thank you. And I think another question for Pieter is that lipid nanoparticle is actually considered as a formulation to deliver, and as you just mentioned, it serve as a targeting, serve as stability, and Drew also actually he is trying to emphasize on the medical equity. And so for medical equity, we are delivering medicine to less privileged countries. So the condition, the preserved condition, and all the shipment condition would be very harsh or very hard to maintain as we normally see in the more privileged country. So, for example, the delivery of the drug, the medical nucleic acid drug could be. Is it possible to develop something that you can deliver? You can make the drug and so deliver in ambient temperature and something like that. Is that a hopeful wish in the field of the nanoparticle?
I would say it is. It's going to require. Part of the problem with the vaccines, of course, was they were developed so quickly that there really wasn't time to say, from the point of developing, say, messenger RNA for coding for the spike protein. But that was in the clinic in 3 months. Normally, pharmaceutical companies would have done a year or more of stability testing and really optimizing the formulation so that it was you didn't have to store it at -80, et cetera, et cetera. So you're going to. I think that the potential for storage, at say refrigerator temperatures is really there. Whether we're going to ever move into a situation where we have room temperature formulations is arguable, but I do think we're going to be able to get to a point where normal refrigerator temperatures are going to be, these things will be stable for a year. Certainly the medicines that we devised using small interfering RNA have that capability. So the lipid nanoparticle is not a limitation. The limitation is probably going to be the stability of the messenger RNA and finding ways to make that, to really optimize that within the lipid nanoparticle.
Okay, thank you. So the next question is for Dr. Wang, and as you were the vice president of Academia Sinica, so I think you can speak for the academia in Taiwan. So what do you think about what the domestic researchers can do in this direction, please?
Yeah, well, Academia Sinica is supposedly the highest academic institution in Taiwan. So our mandate is to do the best basic research as we can. However, I think time has evolved, and we realize many cutting-edge technologies have many applications, and Academia Sinica should also look into those possibilities. So Academia Sinica now, under the leadership of President James Liao, we of course also look into many cutting-edge technologies. For example, quantum computing, and also there are many, like the superconducting research 2 weeks ago or a week ago. Often you can do it at room temperature. So that kind of advance that we should not overlook. Now, certainly in today's topic, messenger RNA technology, vaccine or medicine applications, it's becoming so important. So therefore, Academia Sinica has assembled a team and working with many of the scientists outside of Academia Sinica as well, try to also produce these pipelines of messenger RNA technologies. And of course, it's already quite well established. So Herbert asked me, do we have any niche that we can get into? Well, I think many of the biotechnology applications requires that you have your own IP. And so can we, the scientists at Academia Sinica or in Taiwan, develop our own IP, which may, well, at least be as good as the current technology or better than the current technology. So in Academia Sinica, under the direction of President Liao, I mentioned about the MBRP, the National Biotechnology Park there. We have an institute called BioTrack, and under the leadership of Han Zhongwu. And so they started to look into this, and I do not know all the whole picture, but what I know is, for example, this lipid nanoparticle, important things is the lipid itself, right? I mean, can we find a better lipid that can assemble into a more effective nanoparticle? We don't know, but we are looking into it. And so yesterday I noticed in your talk, your group has synthesized over 150 different kinds of lipids and try to look, each lipid may have a good application in certain area. But for messenger RNA delivery, can we find a better one? So Hanzong told me that we have a chemist at the Institute of Chemistry. He has synthesized many of them, and we found indeed could be some better ones, and it has been biologically validated, and so that's the direction we're going. We try to see whether or not we can develop our own IP, and then working with the IP of all of you distinguished guests here. And furthermore, we are also building this GMP, kind of pilot GMP plant here, so that we can test this, whatever technology we have on site. And it doesn't have to be a large GMP facility. And so this is also one of the direction we are going. And of course, Academia Sinica is important in talent training. So we will also educate hopefully, young scientists to get into this area. So that's the kind of things we are doing. Of course, Taiwan, maybe later, Yijuang can mention more about at the national scale, what are we doing? And one of them, I know Herbert is leading the charge to develop this CDMO facilities. And so Taiwan, overall, we are assembling all the components together and try to push this messenger RNA. Last, one thing I want to mention that Drew mentioned that you are helping a number of countries in setting up those GMP facilities and so on. And since we are also doing that, it might be useful or important that you guys can help us in this area. Since you are the Tang laureates now, so you have the application to help us. Huh? So perhaps we can invite them as some kind of advisors in the messenger RNA technology development in Taiwan. And that would be, in my opinion, very appropriate and very important to us. Thank you.
Pieter?
Speaking for the rest of the laureates, this is something we would very, very much like to do. So.
Yeah, although we repeatedly heard about messenger RNA technology, but indeed the application of the LNP start with the smaller nucleotide. So we know two major companies because they show their power or their help in the pandemic. One is the Moderna, the other BioNTech. Then both of them use messenger RNA platform. But in fact, there are companies like Alnylam or Ionis who use AOS or siRNA as the therapeutic. And so nucleic acid is the role, and nucleic acid plus lipid nanoparticle is the two major players that make this nucleic acid new modality possible. Okay, so well, really, thank you for your your commitment, saying to advise us. Actually, we can, as the new CDMO, the company or facilities established, we can actually help a lot of company or country to manufacture the needed quantity and the quality of the nucleic acid drugs. Okay, and the next question is for Ho, Dr. Sara Ho. And so from the industrial point of view, what's your suggestion to our colleagues in Taiwan and possibly how to collaborate with international partners?
Yeah, thank you, Dr. Wu. As I mentioned earlier in my introduction, I have overseen many new product development and also new product launch. There is a lot of collaboration, and there is a huge effort and the resources and the time consuming. I think we can see the evolution of medicine from target, small molecular, biologics to immunotherapy. Currently, we are working toward the cell and the gene therapies. So mRNA drugs are definitely playing very critical roles. So if we would like to develop mRNA drugs in Taiwan, I think the first important thing is to find clinical unmet needs and start the development. For the mRNA drugs, there has been more than 500 clinical trials in over 20 disease categories. So if we would like to develop mRNA drugs, I think either can be the identified gene in rare disease or markers like a TAA or TSA in different kinds of cancers. I learned from these three laureates. I think it's a tremendous effort and over years. Moreover, the current industrial trend is to build strategic partnership. We need to engage under the current competitive environment and focusing on the speed of the development. So we need to engage the global experts, update the regulation requirements, conduct the clinical trial with the best practice, and finalize the R&D processes. I think a good example, outstanding example, would be the development of COVID-19 vaccines. I think the several outstanding companies were working together with government support to accelerate the product launch and to change the world. I think just as Mr. Wang mentioned, hopefully Taiwanese manufacturer could reach out to you as well and to collaborate with all of your experts and to initiate our research and manufacturing in Taiwan. Last but not least, the development of new medicine consumes a lot of resources. Therefore, we need to cooperate across companies and countries through a collaborative mindset and exploring each other's niche. If the government would be the bridge between academia and the industrials by providing comprehensive support, including platform, update regulations, and funding to strengthen all these connections, I believe Taiwanese biotech companies would be able to devote their energy and resources to development in the breakthrough therapies and bring the hope to the patients, ultimately shining internationally. Thank you.
Okay, thank you. Yeah, sure. Shining internationally. Okay, so shining actually could be responsibility of many sectors, and one of the most important sectors is the government. So the next question is for Dr. Chern, as the Deputy Minister of the NSTC, and what is the department ministers, the ministries, the directions in guiding the development of the nuclear acid medicine, please? Thank you.
Yes, excellent questions. This is a question that we've been asking ourselves. Are we taking the right strategy? I totally agree with Sara that there are many important steps that we have to take care of. Before we address this question, I need to actually highlight there are two crucial advantages in Taiwan that we think would be helpful. The first is that Taiwan has a robust healthcare system that covers nearly the whole population of Taiwan, that result in one of the largest healthcare database. And also, in the past decades, we have collected nearly 700,000 genomic sequence that could be also linked to medical record. So if we combine these two factors, this would be a really good basis for big data analysis, to look for genes, to look for unmet need. So that is our base that we're hoping we could focus on that. And at this time, that we have supported approximately in the past 3 years, about 40 projects. Now, like Drew mentioned in one of the banquets when we chat, that this is still pretty early in Taiwan. So in order to facilitate that, NSTC recently supported 2 core facilities that provide crucial materials and also laboratory scale platform. Now, these 2 core would provide, for example, the initial design and the productions of mRNA, the productions of LNP, etc. When these drugs are ready to move from preclinical to clinical, we are also hoping in Taiwan there are at least 2 to 3 GMP grade facility, including Academician, including National Health Research Institute, that could provide small scale GMP grade mRNA and also probably nucleic acid for clinical trial. Now, for clinical trial, we also have an organization, that part is pretty strong in Taiwan, that is called TCTC, Taiwan Clinical Trial Consortium. NSTC has been supported that for over a decade. So at this time, if we're ready to do that, this is an ecosystem we are trying very hard to develop. And if later we have time, we would appreciate feedbacks and comments and any suggestion that would make this better. Thank you.
Okay, thank you. As I think Pieter, in one of his slides, shows 3 generations of the chemical modality, right? That's yours. Small molecule, biologics, and then nucleic acid. So I think I use the same kind of analogy in many of my public presentation, is that as the new chemical modality for medicine emerged, actually that will create many opportunities, including supply chain, value chain, etc. So in terms of business, it provides a lot of opportunity, but that do not suppress the old modality from emerging into the second generation. For example, like antibody. Antibody used to be a naked antibody, but now everybody is talking about messenger RNA. But actually, in the field of antibody, actually you see the emerge of bispecific antibody, antibody drug conjugate, etc. And for the even for the older modality, small molecule, you think there's no future, but it's not the case. You can see the new technology that protect. So emerge use the novel mechanism to develop. So you can see the new modality emerge, and you see the second generation of the old chemical modality. So I think nucleic acid drug provide a huge opportunity for the industry or for the scientists to pursue. But don't forget about the old modality. But the thing is that, as mentioned, that already 500 clinical trials, nucleic acid-related clinical trials are ongoing. So that means actually a lot of front runner is there. If you join now, it's not late runner, it's a late runner. So actually, we have to catch up very quickly. So maybe another question for Dr. Chern is that have we inventory the Taiwan's academia as well as industry, the players in this field, Dr. Chern?
Like I mentioned, at this time that in academic society that people are working on rare disease. Sometimes there are mutations, and maybe could do enzyme supplements type of assay. But for the industry's part, my understanding is that they is at least one antisense oligo drug for the eye disease. It's been developing. So the antisense oligo part is also very important. And then, like I said, I think we need to do more promotions to actually get the whole ecosystem ready. So all the scholars, they dare to enter, and also probably be able to find the unmet need so that we could actually not become the late runner, but more in front, yes.
Okay, thank you. So next we have the local or domestic panelists to ask question to have the question to the laureate. But before that, I would like to ask one more question to Drew and Katie. Is that Pieter mentions that the improvement of the lipid can only improve the stability of the messenger RNA drug to some extent, but the essence is actually to make the stability of messenger RNA more stable, so that it can be delivered at the more easy way. So what's your opinion about making the stability of the messenger RNA more stable? Maybe Katie first.
I can say that we talk about like siRNA, which can be injected to the blood. I have to say that's really not RNA. The hydroxyl is missing because these two fluoro or methylated. So it is stable because that critical hydroxyl, which is present in ribonucleotide, is not there. So that's why, you mentioned that you can keep it in room temperature, but because I don't consider siRNA an RNA, but so the messenger RNA, even if you would put in a sterile condition and put in an incubator, it will degrade. You don't need enzyme to degrade RNAs. If you get RNAs, then in no time it will degrade. So I question that how it could be altered, the stability, but I think that it still is better if it's frozen, because we can talk about that. Okay, you can keep it on room temperature. Yeah, it will be, 1 day. And minus, you keep in minus, because you have to, when you say the temperature, you have to say the length of the time you can keep it there until it degrades. So if you put in a -80 with the LMPR, you know, maybe forever, because we had 6 years we kept such condition, and when we took out, it was just as active. So then you don't have to go through your inventory and throw away after, you know. So that, I don't know. As I work with RNA, I question that, you know, in high temperature, you can keep the RNA.
Okay, the same question for Drew, please.
So as Katie mentioned, RNA has stability issues that we don't know how to address right now. But there are other technologies that can help this. So what we've made a vaccine in Thailand that's in clinical trials. We've been able to increase its stability at 4 degrees to 6 months, which is the amount of time you need to distribute to an entire country. But going forward, you can lyophilize, you can dry RNA, and it's stable at room temperature for a very long time. You can't mix that with LNPs at a bedside, but we've developed other delivery technologies that can be mixed at a bedside. So if you have to have room temperature stable RNA for vaccines and for therapeutics, that's possible. We continue to develop new technologies to increase room temperature stability. We've been able to freeze dry LNPs with RNA in them using an inexpensive non-lyophilization procedure. So we keep developing new technologies that will allow room temperature stability for long periods of time. Someday we might be able to address RNA stability issues, but that's a long-term goal. In the short term, we are able to make room temperature stable RNA vaccines and RNA therapeutics. And those are things we continue to develop.
Yeah, thank you. It's very nice to know that. Do you have anything to add, Pieter? Or?
No, not really. I mean, I think, as Drew said, that technology is the same in many, many fields. Once you get something that works, then there's this incremental progress that you always see being made, 10% better every year, whatever the, and so you're going to see one way or another, we'll figure out ways to, you know, to maintain stability of the mRNA, certainly improve the potency of the lipid. Now, we're in very early days of this. You know, I wouldn't worry too much about being behind. The whole technology is really in its infancy. And those you know, those advances as we make these things, you know, less expensive, make them more quickly, make them more potent. This is going to happen, the same as in the semiconductor industry and every other industry that you care to name. And so it's certainly not too late. I mean, we're in the very, very early days, and there's so many diseases to go after, so many applications. It's just very exciting. Is really, really what it is.
Okay, thank you for your information. That tissue-specific targeting is also is one of the major interests in this community. So next, I will invite our panelists to raise question for our laureate. Maybe first from Andrew.
Yeah. My question is directed to Drew. Now, mRNA, of course, is a biological molecule, and it will enter our body and supposedly do something there. So understanding the kind of physiological role of those molecules in the body is important. Now, I think I will ask two short questions. One is related to so-called safety. Now, as you may not notice, that outside the auditorium here, there were protesters out there raising banners and saying that, well, long-term effect of this mRNA vaccine. Do we know? Because it just came out of the market so shortly. And so how do we know 10 years from now, 20 years from now, what's going to happen? So that's a long-term safety or its side effect may be an issue that I know it's quite safe. I mean, it has been administered to over 1 billion people, but mostly safe. But Chief Wang, our president, former president, he was so into the messenger RNA vaccines, so he took 6 shots, and because he thought the more the better. And but in the end, the 6th shot, he got a rash all over the body, and it's quite serious. He had to enter the emergency room, and he's still not fully recovered. So there's something we still don't know. Okay. The second question I have is, now messenger RNA vaccination here seems to be so powerful. Do you think it's going to wipe out the protein vaccines, and we shouldn't do any more protein vaccine? That's my 2 questions.
So for safety, the first thing, as was mentioned earlier, RNA is a prodrug. It makes a protein that's the actual drug. The RNA is gone from your system in days. So the long-term effects of the RNA are very short. The RNA is completely natural. It's identical to the RNA in our cells. It's digested in the same manner. It doesn't lead to byproducts that have long-term adverse effects. And it's gone in a couple of days. The long-term effects of the protein are different. So the rash is probably from the spike protein that's induced the T-cell response that resulted in a rash. That could be identical to the rashes we've seen with adenoviral vaccines, with DNA vaccines, with protein subunit vaccines. So I'm not sure that's an adverse event of the RNA. It was caused by the RNA vaccine, but it's an adverse event of the spike protein being given 6 times to a person. For vaccines in general, the first 6 weeks, you see 95% of the adverse events of the vaccine. After that, it's incredibly rare. For RNA vaccines, as I said, the RNA is gone in days. The lipids, so there's 4 lipids in the lipid nanoparticle. 2 are completely natural. They're identical to the lipids that are in your cells. There's 2 other lipids that are foreign. Those lipids are cleared from the body quickly. It varies by which vaccine, but within days, those lipids are gone. So it's hard to imagine you're going to see an adverse event 5 years later from the RNA and the lipids that are gone in a couple of days. The vaccine itself can have long-term events, but that's the same for all vaccines. So I don't see as big an issue with long-term adverse events. I think the problem is, and the protesters that were there aren't scientists who study RNA and know RNA biology or lipid biology. They read social media that says, oh, well, the RNA is going to change your DNA and give you cancer in 20 years. That can't happen. It simply cannot happen. That's nonsense that social media spreads. I've seen every imaginable type of nonsense. I've seen people go to Congress and say the vaccine made them magnetic. I've seen people who say that Bill Gates is putting chips in the vaccine to track people. You know, if you notice, they sit there on their cell phones and they type this in. That cell phone tracks everything they do, everywhere they go, every word they type in. Why does Bill Gates have to put a chip in the vaccine when they're walking around with cell phones that do all of that? So those protesters need to get their facts straight in order for them to complain about the RNA vaccines. I don't know if RNA vaccines will replace all other vaccines. It's too early to know. The advantages of the RNA vaccine are that you can put many different antigens in a single vaccine. So I say this, and pharmaceutical companies always get upset, but it could be in the future, instead of bringing your child to the pediatrician every month, 2 months, 6 months, 12, 16, 18, 24 to get vaccines. And kids are getting 20 or so vaccines over and over and over throughout their childhood. In the future, it might be possible to make an RNA vaccine that has all 20 antigens in one vaccine. And you get it at 1 month, 6 months, 2 years, maybe 6 years. These are just guesses. But RNA could greatly simplify the vaccination of our kids in the future. Will all vaccines be replaced by RNA? I don't know. It's possible. I kind of doubt it, but it's possible.
Thank you. I feel safer now. I can sleep. Yeah.
Yeah, I think as you can see in the bus, almost every single person holding a cell phone like this. So it's more easier for them to track people using the cell phone rather than the vaccine. But one thing that raised my curiosity is that the BioNTech and the Moderna actually start with the vaccine against the influenza or flu. So actually, it is still ongoing. It's resumed activity already, right?
So we, at Penn, we had 3 clinical trials that were started before COVID ever hit for flu and for HIV. So RNA LNP vaccines were already in clinical trials. Moderna did clinical trials a few years before that. So the COVID-19 vaccine didn't come out of nowhere. It was being developed. It was being used for flu, for HIV, for many other diseases before COVID ever hit. It just happened that it was the perfect platform to develop for COVID because it could be made so quickly and it was so potent. And that's proven itself. But the vaccines, the therapeutics were developed before COVID. They continue to be developed. They're going to continue to be developed. We've got a bunch of different pathogens, including norovirus, C. diff, malaria, TB, dust allergies, dust mite allergies, peanut allergies, autoimmune disease treatments, genetic therapies. All of these are headed to clinical trials. So the future is going to be exciting for RNA LNP technology.
Okay, thank you. Maybe Sara, for your turn.
Yeah, I do have 2 questions. The first one is for Dr. Weissman and Dr. Cullis. As a Taiwan-based manufacturer focused on liposome, it's really exciting to see how much progression does the LNP so far. And even I learned from yesterday from Weissman regarding the target LNP can reach to the target organ like lung and the liver and even stem cell. So as a Taiwan-based manufacturer, if we want to go further, what are your advice? And the second question is for Dr. Kariko. If we would like to develop RNA drugs in rare disease or oncology, especially for high prevalence in Asia region or Chinese population, what are your advice? Thank you.
Yes, just with regard to the, I think that the Taiwanese liposome company has been focused mainly on delivery of small molecule drugs. Is that correct? Yeah, the chemotherapeutic drugs, right? Yeah, and there's a lot of lessons that you've learned in that process. You know, it's like Doxil and other formulations that are very valuable actually for the delivery of nucleic acid-based drugs. And so you can build on that expertise, and not just from the formulation, but also from the from the behavior in vivo. One of the things that we've been in that area for a long time, And one of the things that is, you don't hear too much about it, but the thing that always kills a drug, or makes a drug not make it through a regulatory authority, is the toxicity of the drug. And that's one of the things you learn with the delivery of, say, chemotherapeutic drugs, is anything that is, if you're not really paying very close attention to the toxicity profile, then your drug is not going to make it. And I think this is a lesson that is going to be a really strong one in the nucleic acid-based drugs. For some applications where you don't need very much of the protein to be made, such as, say, in a vaccine, then the toxicity of the carrier, et cetera, is not so much of an issue. But for many other applications, toxicity is going to be a primary concern. When we moved Onpattro, the siRNA formulation, into the clinic, we had, in mice, we had a therapeutic index of 1,000. In other words, we could give 1,000 times higher dose before we could see a physiological effect at one dose, but we could go much higher before we saw any toxicity. And I think with a lot of the mRNA-based drugs at the moment, we're not operating anywhere near that range. I think it may be a therapeutic index of 10, but we got to get that up. It's going to have to be in the 100 range before you have really widespread clinical adoption. So that's one area that I think is going to be really important to getting these drugs generally accepted.
I completely agree with Pieter. I think what's important, so for the audience, a liposome is a lipid, a bilayer filled with an aqueous fluid. A lipid nanoparticle is a solid lipid. So the interior is lipid and RNA. The exterior is a membrane that encompasses it. That's the big difference between a liposome and a lipid nanoparticle. They both have usefulness for a variety of applications. As an infectious disease division doctor, liposomal amphotericin is a life-saving drug. Amphotericin is an incredibly toxic drug. When people put it into liposomes, it became much easier, much safer, and much more effective. But I think you have to choose your delivery platform. And I don't think liposomes are ever going to be great for delivering RNA compared to a lipid nanoparticle. But I think it's important to use your knowledge of lipids to develop the right delivery technology for every therapeutic you want to deliver. And for me, that's where I would tell you to investigate.
Yeah, many thanks. So maybe one further question for Dr. Weissman. Consider the targeted LNP. Would you recommend, if we target liver, because that's high prevalence in Asia region, if we started from the targeted LNP, would you recommend to do this way?
So LNPs naturally target the liver because they bind a protein known as ApoE, and ApoE targets liver. The issue that most people run into with LNPs is the IP, is the patent situation. And what a lot of companies have done is they try to develop their own LNPs that avoid prior patents. I have nothing to do with that. I'm an academic researcher. I do whatever I want with anything I get my hands on, and I let the patent lawyers worry about whether we can license it or use it. You'll have to talk to your patent lawyers on how to develop your own LNPs that you can make and sell and develop. Liver is the obvious easy target because LNPs go there. On Onpattro, the numbers I've seen is that it delivers to 95, 98% of hepatocytes and delivers drug. That's incredible efficiency, and that's why patisiran on Onpattro works so well. You'll have to figure out your own kinetics and development if you want to do your own liver-targeted LNPs.
Yeah, thank you so much.
So considering, I am not an oncologist, I am not an MD. Drew is MD, and I am a biochemist. But, you asked me oncology question and related that what kind of cancer is more prevalent here in Asia than other place and what could be done. So one of the significant difference is lung cancer. And lung cancer, those who are not smoking, because they are deep frying. Okay, so that cause also the burn grease product inhalation of those can cause cancer. So that maybe prevention should come first, and for cancer is vaccination is possible only those type of cancers where accumulated mutation, neoepitopes are formed, and not all of the cancer. There are cancer where there is actually no mutation at all. So you have to learn that. And of course, there is not just vaccines are there, which, but there are also messenger RNA code for different cytokines, for example, which can which can be injected to certain tumors close to the cell surface, like melanoma, head and neck cancer, and then making the cold tumor hot, and then inviting those immune cells, and when circulating, they can eliminate metastatic and also that other technologies combined, like, there could be a T-cell therapy or CAR T-cell, for example, and to make sure that the T-cells will not still divide, so that given together with the vaccine. So there are combination of different kind of technologies, what is at the forefront right now. And of course, antibodies, because, in some cases, the cancer cell has on the surface a protein which is specific for that type of cancer. But these kind of proteins are rare. So that's why, for the cancer vaccine, has to be, cell-based and not for much antibody. But antibodies also present, and I mentioned in my presentation that Claudin 6 and Claudin 18.2 is the two of those that is targeted by antibody and bispecific antibody coding mRNA at Biotech clinical trial.
Okay, thank you. So we have Dr. Chern to ask question, please.
Okay, I would like to start by thanking Pieter. Yeah, I totally agree with you. I think there are so many diseases without cure. And also, like Drew mentioned this morning, there are diseases, for example, ALS, that require more than one messenger RNA. So in the future, those would be really very exciting. And I think scientists in Taiwan should feel like we still have excellent many opportunity to get ahead. My question, of course, is the case for mRNA vaccine is a historical event. But now we enter a very exciting era. And how can we do better? How can we more efficiently move the findings in the laboratory into industries? Any feedback or suggestions would be highly appreciated. Thank you.
I can say something like in the United States, so they gave out a grant to academic setting and then included the phase 1. Yeah, it's for so that the scientists who are doing the basic scientific experiments, they also have to think about what will be the phase 1 trial. And so that would help, because the grant will include the phase 1. So it helps, because what happened prior to that, they did the basic science, and then they didn't think about how it will go to the clinic. But if you include, and support, financial support, you are government here, yeah? So that government should do.
So I think what's important, I mean, United States has nearly 700 million people. Mainland China's over 1 billion. Taiwan is, what, 23 million? So the relationship is you're going to have many fewer scientists. I think what's important is you need to identify the scientists who are doing novel and interesting things with their particular diseases, and then work with them to figure out what RNA therapeutics do they think they want to or needs to be developed. You need to take the richness of your resources currently. Going out and trying to make another COVID vaccine and competing with Moderna and Pfizer BioNTech doesn't make sense for Taiwan. But you need to find your excellent researchers who are leaders in the field and help them develop novel RNA therapeutics. And I think that's where Taiwan will really excel.
Thank you. Novelty. I would also take that down. Thank you.
Yeah, I think the other thing is to really smooth the path. If people want to, they're working in their university laboratory and they want to get they want to explore the therapeutic opportunities of their discoveries. At least I don't know what the situation is in Taiwan, but I can tell you the situation in Canada and actually through much of North America. It's it's an awkward process. Bringing something from your laboratory and then getting it into a company environment. And so the degree to which you can facilitate that, and actually in many ways you have to encourage universities, this is part of their mandate. They don't normally like that. It's not a normal part of a university's mandate to make sure that they're commercializing discoveries made in their laboratories. But I think it should be part of the reward structure for, say, university professors that they do this sort of thing, and I think that would help a great deal in getting things, the discoveries into commercial applications.
Yes, thank you. Better ecosystem and also better awarding system. Thank you.
Yeah, okay. Thank you for all the questions. And next, I would like to read a paragraph prepared by my staff about the chronicle of the development of the messenger RNA vaccine during the pandemic. So on January 10th of 2020, the SARS-CoV-2, subsequently called COVID-19, is published. So again, it's January 10th of 2020. And January 13th of 2020 is the sequence or the ultimate drug is designed and later named as the messenger RNA 1273 sequence. So 3 days after the publication by the with the aid of AI and the big data, so they already in 3 days they make the final product sequence. And with the incredible speed that they initiate GMP production of mRNA 1272 on January 14. It's 1 day after the design. And manufacture the clinical drug on February 4th. How many days? 20 days. Start GMP manufacture. And submit an IND application on February 20th. Is 16 days for the preparation. You just cannot imagine the preparation of all the IND, the application material in 20 days. It's incredible. And commence phase 1 clinical trial on March 16 is another 30 days. Okay. Eventually, on December 18, they obtained emergency use authorization EUA from the United States, enabling clinical use. Today, the messenger RNA, the mRNA 1272 vaccine has been administered in 88 countries worldwide, generating a staggering 36.5 billion in revenue for Moderna from 2020 to 2022. I think it's as big as the BioNTech anyway. So from this material, we can inventory what is needed. One is to develop this is, of course, science itself. You have to discover medicine also, and AI, big data, and manufacture, and a good regulatory system, efficient regulatory system, and also clinical trial environment, and etc. So I think by inventory, these elements that needed for the development of the messenger RNA, we can actually see that Taiwan do have the strengths in developing in these directions. Oftentimes I was asked about what is the advantage of Taiwan to develop the biotech. My answer is there's no advantage. We only have strengths. Okay, advantage means superior over others. I don't think we have anything superior or any single thing superior than others. But put together all the strengths, actually we can have the opportunity to prevail in the competition. And also from the material I just read, I actually feel a little bit worried about the older chemical modality for the vaccine development. The subunit vaccine seems to be heavily strengthened by the new technology. What do you think that's true?
So I think what's made subunit vaccines better nowadays is the adjuvants. In the old days, subunit vaccines were given with either no adjuvant or alum, which is a terrible adjuvant. When that was done, it made an antibody response. It made very poor T cell responses. For tetanus, the only thing you need is an antibody response, so it worked fine. For COVID-19, the T cell response is what gives you long-lived protection against serious disease and death. So the T cell response was critical. If you made a subunit vaccine with alum for COVID-19, it doesn't work. It actually makes things worse. The Novavax, they had a very good adjuvant that made their subunit vaccine much better. So subunit vaccines are not dead. I think they're very important. They continue to be developed as new adjuvants are developed. They are expensive, and that's something you have to consider when making a subunit vaccine. But subunit vaccines, the Novavax is as good as any of the RNA vaccines, and I think they should continue to be developed with new and better adjuvants.
Okay, thank you. So actually, my original statement is still valid, that the emergence of the new modality will not actually kill the old modality. The old modality still can survive in some particular field. Okay, thank you. So we have a lot of questions from the audience. So although with the Taiwan, we still have the eager to come here. So I just picked some of them. I just read the first question is I have three leading questions. First three questions, and those questions already been answered during the conversation. So the next one is how do you think on the siRNA application in terms of cancer therapy, since messenger RNA already can play many roles in terms of cancer therapy. So maybe this one is for Drew.
So it sounds like they're asking about siRNA for cancer. I think, and we've got a couple programs that are combining different therapeutics. siRNAs are good at knocking down proteins. Mainly it's directed at oncoproteins, oncogenes in cancers, where if you can get rid of those effectively, you reduce the ability of cancer cells to divide. Nothing is 100% delivery, which is the big problem for cancer. In order to cure cancer, you have to kill 100% of the cells. So what people are looking at is combining siRNA directed at oncogenes to antigens and vaccines to stimulate immune clearance, to checkpoint inhibitors that boost the immune response, to ASOs that alter the shape of the RNA and the types of RNAs. So for cancer, I don't think a single therapy is ever going to be effective. I think you need to combine many different therapies together. And that's where adding siRNA to mRNA to checkpoint inhibitors to RNA-encoded antibodies, all of those together, I think, need to be looked at to effectively treat cancer.
Okay, thank you. So the next question, I think already be answered, but I just read it anyway. It's about what is the issue yet to be addressed in the field of LNP and the messenger RNA. I think Pieter mentioned about that already, right? So we'll skip this one. And the next one is how, I think this one also answered by Drew. How can we solve the immune response caused repeated dose of the LNP messenger RNA injection? You just mentioned it's not really because of the messenger RNA, or it's because of the adjuvant, not adjuvant, the target protein itself. Okay, yeah. I have to go through this. I think most of the question has been answered throughout the discussion. So I think, again, I would like to, in the matter of the time, I think it's about time to conclude this wonderful discussion. So I'll make a brief introduction, a brief summary of this panel. Is that the messenger RNA technology has made a big advance in the medicine area in terms of the new modality, and used and already proven to be safe and useful and efficient. As I just read, the material is so quick from start from scratch, and actually a lot of new drug development has been used in this field, and so. Not only messenger RNA, it's nucleic acid as a new modality itself. So we can have siRNA and antisense oligos together. So I think, again, the emerge of the new modality create a lot of opportunity, yet it will not kill the old modality, okay? Again, thanks all the panelists for the wonderful and insightful discussion, and also thanks all the audience to pay your attention. I think I would like to conclude this panel discussion. Please give our panelists a big hand. Thank you. Thank you very much. And thank you all. Thank you.
Thank you very much to Dr. Wu for moderating the session for us. Actually, Dr. Wu, our moderator, have prepared a special gift for each one of our panelists to express his appreciation and gratitude toward our distinguished panel.
And before I present it to you, I would like to make a brief introduction about the gift. It is not a commercial one that we bought from the street. Actually, it's designed by myself and handcraft, and make the stem myself, and then print it on the souvenir, okay, on the glass.
Well, thank you, Dr. Wu, for preparing this very special gift. And thank you once again to our 3 laureates, Dr. Kariko, Dr. Weissman, and Dr. Cullis. Thank you for coming all the way to Taiwan and for your generous sharing. And also, we would like to say thank you to Dr. Wang, Ms. Ho, and also Dr. Chern for contributing in this inspiring discussion. Thank you very much. And now we would like to invite you to take a group photo together on the stage to remember this special event. You can put the gift bag on the couch. Also, we would like to invite Dr. Yan and Dr. Zhang to please join us for a group photo on the stage. Thank you very much. Thank you, everyone. Please look into the cameras in the center together, and don't forget to smile. And now please look at the cameras on your left. We have some cameras here on your left. And the cameras on your right. Thank you very much. How about we turn to the center again? Thank you. Beautiful photo. Thank you very much. Thank you once again to all our panelists for your inspiring discussion. Thank you. Ladies and gentlemen, this concludes Tom Price Taiwan Biotechnology Forum. Thank you very much for joining us on this Typhoon Day. Please don't forget your personal belongings when you leave, and we wish you a good day. 各位来宾，唐奖台湾生技奖到这边圆满的告一个段落，非常感谢您今天的热烈支持。离场的时候，请记得您随身携带的物品，并且归还您所借取的同步口译接收器，取回您的证件。另外，今天有停车的来宾，也请在场外的服务柜台来领取停车时数券。祝福大家平安顺心，谢谢。